Hi, this is GCSE 9-1 Crammers. It's never too late to start revising, and this way you can do it anywhere. Listen to these questions as many times as you need until the answers pop into your head. Today we're revising biology, topic 1a.2 Microscopy, covering light and electron microscopes, converting between units, the magnification formula, and expressing answers in standard form. Types of microscope: the light microscope and the electron microscope. The light microscope uses light and lenses to magnify an image of a specimen and let us see individual cells and larger subcellular structures. The electron microscope uses electrons to form an image. This lets us see much smaller subcellular structures in greater detail. This is because an electron microscope has a higher magnification and a higher resolution, giving a sharper image. Exam-style questions. How does a light microscope form an image and magnify it? The light microscope uses light and lenses to form an image and magnify it. Compared to a light microscope, how would an electron microscope form an image? An electron microscope would use electrons instead of light to form the image. What is the difference between images seen under a light microscope compared with an electron microscope? Using a light microscope, you would only see larger subcellular structures, whereas using an electron microscope, you would be able to see much smaller subcellular structures in greater detail. What is the advantage of using an electron microscope? An electron microscope has a higher magnification and a higher resolution. What does it mean if an image has a higher resolution? The image would be sharper. Size of organelles. List the organelles from largest to smallest. Nucleus, chloroplast, mitochondria, ribosome, lysosome, centrioles, nucleolus. Which cells would you be able to see using a light microscope? Nucleus, chloroplast, and mitochondria. Which cells would you only be able to see using an electron microscope? Ribosomes, lysosome, centrioles, and nucleolus. What type of microscope is needed to see ribosomes and mitochondria? An electron microscope would be used to see ribosomes and mitochondria. In the above question, the answer would have to be an electron microscope because you could not see ribosomes using a light microscope. Converting between units. Take time to learn this, it's important to remember. For magnified images, they will be measured in millimeters being the largest, then micrometers, and nanometers being the smallest. When converting a larger unit to a smaller unit, you always multiply, and the answer will get larger. So converting millimeters to micrometers, you multiply by 1000, and converting micrometers to nanometers, you multiply by 1000. When converting a smaller unit to a larger unit, you always divide, and the answer will get smaller. So converting nanometers to micrometers, you divide by 1000, and converting micrometers to millimeters, you divide by 1000. So, if you get asked to convert millimeters to micrometers, you remember millimeters are larger than micrometers, so you would multiply by 1000. Convert nanometers to micrometers. You need to remember that nanometers are smaller than micrometers, so you would divide by 1000. Convert micrometers to millimeters. Divide by 1000. Convert micrometers to nanometers. Multiply by 1000. Learn to use the magnification formula. What is the formula equation for magnification real size and image size. The equation would be magnification equals image size divided by real size. You can also use the equation triangle. Cover the one you want to find with your finger, in this case magnification, and use the equation you can see, image size divided by real size. What is the formula to find the real size of an object? Real size equals image size divided by magnification. If you are using the equation triangle, cover real size, as it is the one you need to find, and use the formula image size divided by magnification. Take time to learn this equation. It's important to remember. Tackling magnification questions. Remember, when tackling any magnification question, draw the magnification equation triangle out to help you. Also, remember to make sure that all the measurements are in the same units. First question. A bacterial cell is 5 micrometers wide and has an image size of 5 millimeters wide. What is the magnification? What does the question tell us? The real size of the bacterial cell is 5 micrometers. The image size of the bacterial cell is 5 millimeters. I need to find the magnification. The first thing I must do is to make sure that all the measurements are in the same units, and I can see that in this question they are not. So I must convert them both into micrometers or millimeters. I choose to convert them both into micrometers. I know that millimeters are bigger than micrometers, so I need to multiply by 1000. So I multiply the image size of 5 millimeters by 1000 to give me 5000 micrometers. Look at my equation triangle. Cover magnification with your finger. Now write out the equation. Magnification equals image size divided by real size. Substitute the figures. Image size 5000 and real size 5. Do the maths to get the answer. Magnification equals times 1000. And that's the question done. It's important that you really know the magnification equation and how to convert units for the exams. Tackling magnification questions. So again, remember, when tackling any magnification question, draw the magnification equation triangle out to help you. Also remember, make sure all the measurements are in the same units. Second question. A cheek cell is magnified 600 times. The width of the image size is 10 millimeters. Work out the real width of the cell and give your answer in micrometers. What does the question tell us? The magnification of the cell is 600 times. The image size of the cell is 10 millimeters. I need to find the real size. Look at the equation triangle. Cover the real size. Now write out the equation. Real size equals image size divided by the magnification. Substitute the figures, image size 10 and magnification 600. Do the maths to get the answer. Real size equals 0.016 millimeters. The question asks for the answer in micrometers. We know that millimeters are bigger than micrometers. So multiply the answer 0.016 by 1000. Giving an answer of 16.6 micrometers. Writing in standard form. In question 2 you were asked to convert the answer to micrometers, but you could have been asked to write this in standard form. What is standard form? It is writing your answer to the power of 10. So how do you do this? Firstly, the first number in your answer needs to be between 1 and 10. This means moving the decimal point after the 1 in this answer. count how many decimal places the pointer's moved. You can see it has to move 2 places to get behind the 1. Therefore 2 is the power of 10 you will use in your answer. The power of 10 will either be a positive or negative number, depending on which way the decimal point moves. This is easy to remember. If the decimal moves left, it will be a positive power of 10. If the decimal moves right, it will be a negative power of 10. So, put a minus sign in front of the power. As you can see, we had to move the decimal to the right. So, we will put a minus sign in front of the power number. So 0.016 millimeters in standard form equals 1.6 to the power of minus 2 millimeters. In this video you've learnt all about light and electron microscopes, using the magnification formula and expressing answers in standard form. The key to exam success is learning, consolidation and keeping this topic fresh in your mind. How do you do that? Repetition, repetition, repetition. If you go over the facts and these equations enough times, you'll be able to answer any questions on this topic confidently in any exam. This is GCSE 9-1 Crammers, and if you like this video, listen to it as many times as you want, and look out, we're putting out a bunch more, so smash that subscribe button, hit the bell icon down below to notify you of new videos coming soon on more GCSE biology topics, and click that like button.